Good morning. I feel like I'm stopping everybody from having such a good time, but it is time to begin. My name is Gordon Studging. I'm the coordinator for this class on bugs and guts. In 1985, when Guns N' Roses was formed, I thought, if I ever join HASP, I'm going to have a class with an N in the middle like that. And it took a while to come up with bugs and guts, but it's about the microbiome. It also took me quite a while to find somebody to talk about this. It's sort of a new topic. It's an old topic, but it's new for our awareness. But I found Ian Cleary at Grand Valley State University. Ian is a native Canadian from Alberta, got his graduate degrees from the University of Alberta, including a PhD, and is now an associate professor of microbiology at Grand Valley. We're very happy to have him here. This is a 3-session class, so we'll see you the next couple of weeks also. Help me welcome Ian Cleary.
Great, thank you very much. So I teach, the focus of my teaching is microbiology, and I work with Candida albicans, which is a component of the normal microbiome of most of the human population. So I don't study the microbiome directly, but I teach a lot of things related to it, and I study an organism that's part of it. So today's session is all focused on really what is the human microbiome? So what is it made up of? Where do we find it? And what are some of the interactions that we have with it, with all of these microbes associated with us? And then the next sessions focus more on some of those direct effects that these organisms can have on us, on our development, to a degree, on our behavior. And then the third session getting more into sort of the disease side. What happens when we sort of lose that balance between the microbes and us? So that's sort of where we're headed with things. So again, we'll start out by just talking a little bit about what sorts of organisms are associated with us, where we find them, because we are a diverse collection of environments. And so we've got a diverse collection of microbes that go along with that. And then again, wrap up with some of the ways that we benefit by having these microbes associated with us. So it's often stated that there are more microbial cells associated with us than there are human cells. So probably 10 times more microbes associated with us on the skin, in the gut, than there are human cells. Now, obviously, in terms of size and volume, there is more of us. There's more human material, but there's a tremendous number of these microorganisms. And when we look at sort of the genetic side of it, there are probably more microbial genes associated with each of us than there are human genes, because again, we've got these thousands, millions of different microorganisms, each doing their own thing. And so there's a lot going on that we can't see, that we aren't aware of. This community is always with us. It's with us, and indeed we'll look at some of the details about that. How do we first acquire this microbiome? And we've got some members that will be with us long term, that are probably always associated with us, but we've got others that are just transient members of this microbial community. And you can imagine that if you are, say, out gardening and you're digging in the soil, you are picking up a tremendous number of microorganisms. You wash your hands, most of those go away, and so they may only be short-term visitors. So this is not just a fixed population that is always the same group of organisms, the same numbers. This is constantly changing depending on what we are coming into contact with, and depending on the changes in our body and what resources are available for the microorganisms. When people study the microbiome, there are some different approaches, and I'm mentioning this because this influences what we know about the microorganisms and some of the conclusions we can draw from some of the studies that have been done. So really, we've got two approaches if we want to know what's associated with us. One is we can culture organisms. We can swab the skin, we can streak some things on a plate and see what grows. And of course, that's where we started. People just sampled different body environments, tried to grow some things, and worked out what was around. E. coli is a common component of the human microbiome, and it works out that it's really easy to grow. And so it's been a model system in microbiology because people could isolate it from humans. It grows really well, it grows really easily. So we've known for a long time that we're associated with E. coli. As we've developed new techniques, we've been able to try and find things that we can't grow in the lab. So there's lots of microbes associated with us that we don't know how to best grow them in a lab setting. So how do we know that they are there? Well, if we look for DNA sequences, that will be an indication that the organism is around. So if its genetic material is present, then it stands that that organism is likely around in that environment. So a lot of the new work that's being done related to the microbiome is based on that genetic profiling. Collect the genetic information, compare it to databases, work out what organisms are present, but there is a pitfall to that. If there's DNA there, again, presumably the organism is there or at least was there. If there's genetic information, it doesn't tell us that the organism is still alive or there is an active population. We just know that at some point there must have been enough cells to leave some DNA behind. So again, it's a powerful tool. It allows us to ask some different questions, but there are some limitations of that. And again, I'll talk about that a bit later. So that's why I'm bringing it up at the beginning, sort of how we get this information and how we learn about these organisms. So the human microbiome contains any kind of microorganism that we can think of. We tend to talk a lot about bacteria, and there are lots and lots of different bacteria associated with us, but there are fungi, like the organism that I study. There are other eukaryotic parasites, things like nematode worms, although those are less common than they once were. There are organisms that fall into the group the archaea. Now, these have traditionally been described as extremophiles because many members of that group we find growing in places like the Great Salt Lake or deep sea vents where things are very hot and we've got high pressure. And so we typically thought of those organisms as being only in those really extreme environments, but it turns out some members of that group live in the gut and happily produce methane and feed off of the things that we are eating. So we've got all kinds of different organisms. There are also viruses. Now, when we see that, think, okay, well, there are viruses associated with us. Sure, we know that we can be infected by viruses, but all kinds of different organisms have viruses that infect them. So when we look at the viral component of the microbiome, yes, some of those viruses are going to be things that infect us. Some of those viruses are going to be viruses that infect bacteria, viruses that infect fungi. And so this is really the first indication that not only are all of these organisms interacting with us, they're interacting with each other. And so if we're trying to understand how this community is functioning, it gets very complicated very quickly. We tend to focus on the human side because we care about things like human health, human function. So how are these things interacting with us? But there's a lot that's happening just between the different members of this population that to a degree don't necessarily involve us directly, right? These organisms doing things to and with each other, all in this environment, which is us. So it's a complicated population. It's quite varied and there's a lot going on. As we started to learn more about the human microbiome, we've also been studying the microbiome of other organisms. And what we find is that when we start to profile and make some comparisons, we do have particular groups of organisms that we find associated with humans. And as we broaden the perspective a little bit, we notice that, well, in blue, we've got things that are associated with humans, but we're starting to see some overlap with organisms we find in other animals. And if we broaden it further, again, we can see sort of some of our characteristic microbes that are also found in, say, other mammals, in reptiles. So again, this population of microbes is really varied. Some of them are going to be unique to us. Some microbes are only going to be associated with humans. They've clearly found a comfortable home and they've adapted to living associated with us, but other organisms are not so fussy and can move, in fact, between different organisms, different animals. So again, to pick on E. coli lives in the human gut, but we'll find it living in the gut of cows. We'll find it living in the gut of lots of different animals. And indeed, it's a microbe that can happily move back and forth between those different hosts because it's really the gut environment rather than a human in particular that it prefers. So again, lots of diversity here. So here we have a person. It doesn't look much like a person, but this is a good representation of a human being. And what this highlights is all of the external points of contact we have with the environment. And virtually all of these surfaces are colonized by microorganisms. So we may think about the GI tract as internal, but functionally, it's an external environment. It's an external contact point. And so there are lots and lots of microbes there, and it's a point of entry, it's a point of exit. So we have to consider all of these different areas as places where we are likely going to find microorganisms. Now, we are varied in our environment. So different parts of the body have very different environmental conditions, and that's going to influence what microbes we find in those different conditions. And we can just look at the colored groups here. So each of these colors represents a different environment and different microbes that are found in those environments. And so we can see that different populations cluster based on the different body locations. There are different preferences for growth. And again, we don't need to worry about the specifics, but we can see quite clearly how these microbes each have their particular preferences. And if we think about some of the conditions that are different in different parts of the body, we can think about pH. So the skin and the stomach are both acidic environments. The skin slightly acidic, the stomach very acidic. So if there are going to be microbes growing there, they have to be able to handle being in an acidic environment. Whereas other microbes that are acid sensitive aren't going to be able to live there. So we're going to have different populations. We've got differences in temperature. The extremities and the skin are going to be cooler than the core body temperature. And that's going to influence the growth of some microorganisms. So some may be only found where it's a bit cooler, only found in those extremities. One of the big influences on which microbes are where is oxygen availability. So some microbes use oxygen to support their growth the same way we use oxygen. Some microbes are killed by oxygen. And so we can only find them growing where oxygen levels are very, very low. So oxygen is a big determinant of the microbial population. So again, we can think about the skin, the upper respiratory tract, the mouth. These are all environments where there's lots of oxygen available. So if an organism needs oxygen, that's a type of environment where we'll find it. But as we get deep into the GI tract, if we're looking at the colon, we're now looking at an environment that's really low oxygen. And so this is where we find organisms that may in fact be killed by the presence of oxygen. So that's going to be a very different population than we find in, say, in the oral cavity in the mouth. Even though the two are linked, there's a physical connection there. The environmental condition is so different, and so the population of microbes is going to be different. Now, having said all of that, there are also some organisms that, like E. coli, that grow really well in all kinds of different environments. So some members of the microbiome are confined only to one spot. Others are all over the place in all kinds of different locations because they can grow well in these different environments. So when we think about the human microbiome, we're not just looking at one overall population. We've got all these subpopulations depending on the different body locations and the different conditions in those areas. So I just want to talk a little bit more about some of these environments, and then what we'll focus on is what's happening in the gut. But if we think about the skin, again, it's slightly acidic, it's open, it's accessible to the air, so there's oxygen. It's also dry. In many ways, it's not a very hospitable environment. Microbes would be, most microbes are going to grow better in areas that are warmer, wetter, have more nutrients. But there are microbes on the skin, and they are varied. So we have, again, a number of different bacteria, and you can see, again, we don't need to worry about the details, but just in any of the pie charts, the different colored sectors are different groups of organisms. So we can see if we look at the skin, we've got a variety of different bacteria. We have some fungi, and in most parts of the body, it's not a terribly varied population, but we get down to the feet, and now we've got a whole host of different organisms that are growing. And again, we've got a difference in condition. It's a bit warmer, it's a bit wetter. We've got some other microbes growing there. And if we think about something, a fungal fungus that's growing on the feet, we can think about the cause of athlete's foot as an example. So again, it's not all about bacteria. There are some interesting fungi out there as well. And there are viruses. Again, some of these are going to be viruses that infect us. Some examples of human papillomavirus here. But we'll find viruses that infect some of the microbes that are in these environments as well. So even though the skin isn't a terribly hospitable environment, we're going to find lots of microbes there. The skin, of course, we come into contact, we're constantly touching surfaces, we're picking up microbes, we're leaving microbes behind. We're also shedding skin cells. So some of the microbes that are only transient members of our microbial population, we're going to lose when we lose those skin cells. So it can be quite changeable depending on what we're coming into contact with and how often we're remembering to wash our hands, for instance. That's all going to influence what types of microbes we find on the skin. The respiratory tract has some microbes that are in common with some that grow on the skin, in particular things like Staphylococcus aureus. I mentioned that one in particular because I'm going to mention it a bit later as well. But if you've heard of, say, MRSA infections, methicillin-resistant Staphylococcus aureus, so that is a member, Staphylococcus aureus, the species, is a member of our normal microbial population. At least a third of the human population is colonized in the nasal passages. It's just a normal member of that microbial community. And again, not surprising perhaps that we find it living on the skin as well as in the upper respiratory tract. We've got some physical proximity there and some similar conditions. As we get further into the respiratory tract, we tend to find fewer microbes down in the deepest part of the lungs, but certainly lots of different microbes in the upper portions. Actually, back up here. So if we think about even something like this, it's a relatively simple view in terms of we've got these broad divisions of the respiratory tract, and we've got different groups of organisms in those areas. But if we think about just the mouth, that is not all one environment. So obviously we're inhaling, so we're constantly bringing in fresh oxygen. But if we look at along the gums or beneath the gum line, we actually find oxygen poor environments. Some of the organisms that cause periodontal disease really do not like oxygen. So even when we think about the mouth as an environment, it's not one environment. It's many different environments, and depending on where we sample, we're going to find different communities of microorganisms. Again, any surface, any point of contact with the outside environment, we're going to find microbes. When we talked about using genetic information, using the presence of DNA to look for microorganisms and to try and understand the community, when we consider the state of the bladder, that is actually a situation where there's a lot of debate right now. You have some people who feel very strongly that this is a sterile environment, that there are no microorganisms in normal situations, and others that suggest that, in fact, there is a resident population. We're just not very good at studying them. And so we haven't been able to grow them well outside of that environment. We don't know much about it. A lot of the debate stems on people finding DNA. Think, oh, there's some DNA here. There must be a resident population of microbes. Well, what if some of those microbes found their way into that environment and then died because they didn't have the right nutrients? It wasn't a very hospitable environment for them. We're going to detect the DNA. It doesn't tell us that there's an active growing population there. So this is one point where there's a lot of disagreement at the moment. And it's, well, at least in scientific terms, the debate gets quite heated. You read some of the papers, they're it's not shouting on the street, but letters to the editor that where people are very adamant in one direction or the other. But again, it just shows you there's a lot we don't understand about these microorganisms. Seem to have frozen. There we go. All right. So the gut is where we find the greatest number and the greatest diversity of microorganisms. And again, we might think about this as one environment, but it's really not. It's multiple environments. We have changes in pH, we have changes in oxygen availability. And so if we think about the population that's in the stomach, and there are a few microbes there, versus the population that's in the colon, those are going to be very different types of microorganisms. Again, some of these may be with us long term. Some of them may only be transient members. And some of them, again, will be confined to only one area, and some will be in a whole range of different organisms. So just if you look at the arrows and pointing to sort of where we find some of these microbes, some of them we notice are only in one spot. And again, we don't need to worry about the details of which region, but you can see some of them are confined just to one area, whereas others, if we look at Streptococcus species, for instance, they're in all kinds of places we sample. So again, when we're thinking about the human microbiome, we've got to think about what's happening in the different areas. We can't just lump it all together in one huge list of everything that we find associated with humans, because they're all in different places doing different things. All right, so how does this all get started? As I was putting this together, I was reminded of something I read years and years and years ago. And it was written in the style of a science fiction exploration. So people land in this new environment and they're finding their way around, and all of these crazy things are happening around them, and they're running into these strange locations. Some are windy, some are dark and enclosed. And it was a story about microbes landing on a newborn and encountering these different types of environments, but from the perspective of what would it be like if we were newly arrived on a human body and trying to find our way around. And we are colonized when we first make contact with the outside world. So before that, no microbes. As soon as we have any contact, then we start to be colonized by these microbes. And we're going to be colonized by whatever we come into contact with. So the air is full of microbes. We are constantly breathing in microorganisms. Happily, we cannot see them, but they are there. So that first breath, we are inhaling microorganisms, and we start to establish the respiratory microbiome. We are touched by someone. Microbes are transferred to the skin, and we start to establish that population. So from the very beginning, we start to be colonized by these microbes. And as I said, some of these microbes might be with us for our entire lives. Again, E. coli, pick on E. coli. It's essentially always going to be around. We're going to be colonized early on. That's one that's going to stick with us. Others may change as we come into contact with new environments. as our physiology is changing. So again, it's a dynamic population. It's not fixed, it's not set in stone, but we will be colonized immediately. There's a lot of interest in understanding the nature of that colonization. What is established first? Why are those organisms established first? And how might that influence future developments? And there's a suggestion that indeed the combination that we're initially colonized with can influence not only the development of the microbiome, but our development. So we're trying to get a handle on how all of this works. Again, some things are fairly straightforward. We know we're going to inhale some organisms, and it'll be whatever is in the air around us. But it also turns out that there's a difference in the initial microbiome between a cesarean section and a vaginal delivery. And part of that is simply because of the contact with the microorganisms. So these represent sampling in the first few days after birth, and vaginal deliveries on the left, cesarean sections on the right. And we can just look at the different proportion in the colors to see that there are obvious differences between the groups. Now, some of these more or less work themselves out in short order. The microbes reach a similar balance. So if we look at the, for instance, this upper portion here, these blue lines, that it really doesn't matter. Those organisms seem to find their balance no matter what the starting point is. But this one group in particular, these Bacteroides groups, are one that have been highlighted as being not particularly abundant with a C-section birth, and which remain less abundant afterwards than we see with a vaginal delivery. Now, trying to go from an observation like that to cause and effect is a whole other question. So we're starting with making some observations. We're getting some suggestions of, well, this means X, but there's a lot where we're still at the basis of, okay, we know there's a difference. How much do we care about that? We're not entirely sure. Because we don't know enough about how this population changes over time, what the long-term consequences might be. We're still at the early days of some of this work, but there's clearly a difference here. There is clearly a difference. And it's not something that's always sorted out in just a couple of days. So the suggestion is, well, maybe this does have a longer term effect. And something else that's been noticed is not only do we have those differences in what is colonizing initially, but when we look at what is showing up, in the case of a cesarean section, we find that, so if we look at the blue bars here, organisms that are commonly found in hospital environments tend to be more abundant. So it seems that there's a greater colonization from the external environment than from the mother, which makes some sense. The first point of contact really is with the atmosphere, with the hospital workers. And so again, the question is, well, does this matter a lot? Maybe. Some of these organisms have the potential to be disease causing. So maybe this is something that we might be concerned about. And so the suggestion has been made, well, can we do anything to influence this? So if we know that there's a difference in that initial contact, and we know that there's a difference then in the resulting population, can we change that? And so the suggestion is, well, what if we swab the mother and then swab the newborn? What if we carry out that transfer? So if the cesarean section is interrupting that transfer of microbes, what if we do it instead? And so there have been a couple of studies, and again, we can see that there are clear differences. Again, even just looking at the patterns, right? Not worrying about the specific groups, we can see that there are different patterns depending on these different environments, different situations. And so this is something that's being investigated. Should we, in the case of a C-section, be inoculating the newborn? Give them some of the same microbial population that we see in a vaginal delivery. On the one hand, that sounds like a great idea. On the other hand, we have to remember that we don't know all the details about how all of these microbes are being established. And if we're just taking a swab, we don't know what we're transferring. We know it's going to be whatever is resident on the mother. We don't know the makeup of that population. And so one concern would be, what if there is something like an active herpes outbreak? Maybe one that doesn't have any visible signs, but nonetheless, we've got viral particles being shed. That can be a really serious infection in a newborn. So we know that there's a difference in the microbiome. We know that we have the potential to maybe do something about it. The how is still an open question. Should we be introducing particular organisms, particular groups, grow them up in the lab? We know what they are. We know precisely where they've come from. Maybe use that as our inoculum? Maybe. This is all still very early days. But we do know that there's clearly a difference in how those microbes are being established. On the experimental side, people have been asking questions, well, does it matter, right? Does it matter the sequence of events when we're colonized, what we encounter first? Does it matter if we see things all at once or we get them one at a time? How does all of this work? Again, with the idea of, could we maybe apply this to the human situation? So this was one study that was done using mice, because we can do this sort of study in mice. We can't do it with humans. And so they started with germ-free mice that were treated with antibiotics to get rid of any bacteria. They were housed in an environment where they were not colonized with all the things that we normally come into contact with. And then they were deliberately inoculated with organisms. Okay, and they were using two situations. So they had their two donors, and they started with donor A and then inoculated with donor B and then flipped it around. Okay, does the order of contact make any difference? That was the question. And it turns out the answer is yes. There were clear differences between these orders. So when they had, there we go, parent A, and they did A and then B, you can see we've got the orange dots here, that particular cluster. When they reverse the order, it's all the green dots, so donor B, then donor A. It mattered. The sequence mattered. It's the same organisms into the same environment, but the order made a difference. And that's because we have to remember the microbes are interacting with each other. They're competing with each other for nutrients. They're competing with each other for space. So the ones that get there first may have an advantage. And it may be harder then for the next population to become established. And so this is the sort of thing that we have to think about if we are looking at, say, a deliberate inoculation after a C-section. We need to know what to put in and what the order is if we're trying to end up with whatever we think is the desirable outcome. So again, it's not just about us. We've got to think about what the microbes are doing with each other as well. Now, we continue to be colonized. It's not all about what happens at birth. Later contact with the environment will also influence what is in the microbiome. An organism that we may pick up a bit later is Helicobacter pylori. Now, this is best known as a cause of ulcers. And famously, the convincing work was when one of the investigators, who was tired of people telling him he was crazy, drank a flask of Helicobacter pylori and then became very ill. But that's what it took to convince people, because the notion at the time, late 70s, early 80s, that a bacterium was the cause of ulcers was not accepted. Now we know that this is actually a major cause of gastric ulcers. it is also, or at least historically, has been part of our normal microbial population. So looking at the nature of the microbes, we think that there's probably been an association of more than 100,000 years between us and this organism. What is changing is, or what has changed in particular, is hygiene. So this is an organism that we don't come into as much contact with anymore. And so when we look in developed countries, the colonization rate is steadily dropping. Fewer and fewer of us are colonized with Helicobacter pylori. And it's because we've altered that interaction with our environment. From the perspective of being a cause of ulcers, this sounds like a really good thing, right? Less colonization. If it's not there in the first place, then it's not there to be able to cause us any harm. So that seems like a great development. There is some suggestion that actually this organism living in the stomach may contribute to regulating acid levels. That in fact, having these organisms around, as long as they're not causing us a lot of damage, may not be a bad thing. So maybe fewer of us being colonized is not really the desired end result. Now, if you put the trade-off of perhaps a little excess stomach acid versus the potential for ulcer development, I think the balance is still very much in the let's not have Helicobacter pylori around. But it's a great example of something that where we're not colonized at birth, we're colonized later, and the colonization is going to depend on what we happen to come into contact with. So some people may be colonized, others may not, and different populations around the world will have different profiles of that aspect of their microbiota, depending on what it is that they have come into contact with. So if we looked in developed countries, we've got relatively low levels. Developing countries, we've got higher levels of colonization with this organism. And a lot of that is through things like sanitation and that direct contact. So as people started to look in more detail at the human microbiome, we had a number of microbiome projects, and some of them were whole body, some of them were focused on just the gut. But the notion was, let's try and establish, let's work out what is part of this population. And then we can start to ask questions about what is the healthy microbiome? What combination of organisms is going to be beneficial to human health? And then potentially, what changes can we try and make in that population to promote health? So that's sort of the premise behind all of this. As I said, the challenge is this is such a complex system. How do we go from individual observations to the functional side to actually trying to influence the host? There are some places where we've had some success with that, and a lot of places where we're still asking questions. But this is where we started, simply by trying to catalog everything that was living associated with us. Now, if we're trying to establish what is the healthy or what is the normal microbiome, how do we do that? We can't pick one person and say, we're going to profile you, and you are the representative human, and that's what we're going to base all of our information on. We've got to take a varied population, because we know humans are varied. We know the microbes are varied. So we've got to take a collection of people, and maybe, again, in this case, we've sampled a few different people, and we've measured what each of them has, and we perhaps try and come up with a common core group of organisms. Okay, we know that everyone has this group, and then there's a lot of variation around that. Okay, that's one aspect. But something else is, what about time? What about changes in the microbiome between different ages? Children, adults, that may look very different. So what is a healthy or a normal population in someone who's 10 may be very different from someone who's 40. And so how much should we be paying attention to those types of changes? How much might this depend on the location we're sampling? Because again, we know different microbes are in different locations. So when we talk about profiling the human microbiome, and what is a normal, what is a healthy microbiome, it can get complicated really quickly. What these studies tend to start with is a population, a group of people who are considered to be healthy. So no obvious sign of active disease. And then we'll sample those people and pool that information together and try and come up with what we think is a normal, a healthy profile of the microbes. And we'll see some of the commonalities, but I also want to highlight again just some of the differences we can see between individuals. So this is the result of sampling 3 different individuals as well as their computer keyboards. And you can see that they're grouped by color. So the 3 different individuals are the 3 different colors, and the keyboard are the closed symbols. The open ones are the actual fingertips. So you can see 2 things. One, of course, is that there are differences even in the skin microbiome between these 3 individuals. So again, trying to come up with what is the normal, what is the standard microbiome for all humans, that can be a bit tricky. But it's also just a reminder of the fact that every time we're touching a surface, we are picking up some microbes and we're leaving some behind. And so we've got to think about that when we're compiling all of this information as well. But what happens when we do collect a whole bunch of information? So I'm going to highlight a couple of different studies that were done. So this one looked at a group of 34 people, and it was focused on the link between diet and the gut microbial population. So it monitored 34 people over 17 days and tracked both their microbes as well as the food they were eating to try and see if they could notice any particular patterns. So that was the setup. And this was a 2019 study, so fairly recent. All right, so the figures look kind of busy, but again, what we're looking at is the pattern. And so we've got our different people here, and then the foods they were eating were grouped together, as well as the biochemical activities of the microbes. And if we look at the food groups, before we look at the organisms or anything else, we notice that with the exception of these 2 individuals who were on a supplement, so something like an Ensure drink, everyone else was eating a rather varied diet. Okay, so again, we've got things like grains in the orange, I got something that's a little more visible, milk in blue. So everyone except those 2 individuals eating a whole mixture of different foods. And you can also see just the difference in kind of the peaks and valleys here, that it changed over the course of the study. So people weren't eating exactly the same thing every day. It varied from day to day. And we can see that there are also differences in the microbes, both within individuals, but also across the group. So the different colors representing different groups of microorganisms. These 3 individuals, there was quite a different makeup of the population. We've got this huge expansion of whatever the yellow group is representing. For other people, if we look in the middle, all of these individuals seem to have relatively similar profiles. So again, this is the beginning point, right? Measure what's happening, measure what's there, and then try and put all of this together to understand what's happening. So if we look a little more closely, so we've got 3 different people here. 2 are the ones that were on the supplement, which is why the lower box is all purple, right? They were on one type of food, one material. The third person, subject 10 there, was eating a varied diet throughout the time frame, and we can see different foods came and went. But when we look at what's happening with the microbes in those 3 individuals, we can see 2 things. One, even in these people who are just consuming the same food through this whole period of time, we maintain a varied population of microbes. We've got all kinds of different microorganisms, and the population changes, right? We've got one nutrient source coming in. It's all the same food all the time, And yet we've got changes in the makeup of the microbial population. So even when sort of the input is the same, the output changes. Some of that's going to be to do with how the microbes are interacting with each other, who's competing with whom, who's producing something that's harming someone else. Some of it's going to be whatever's going on with our physiology, okay? Because it's not just about the nutrition, it's what else is our body doing, what else might we be doing to the microbes. But then the other thing that's interesting is that if we look at the person with the varied diets, we can see that huge changes, and in particular, the authors note that strong association between weekends and the consumption of eggs, which are these two spots here. So even in the course of this time frame, there were some dramatic changes in diet. We actually notice that the makeup of the microbial population is relatively stable. So here we've got all kinds of different foods coming in, but for any one of these categories, it's fairly even, which is what this graph is getting at as well, that we've got, if we have a diverse diet, we actually have a more stable microbial population, which seems backwards at first. You'd think if we've got all kinds of varied foods coming in, that the microbes would be changing constantly. I think really this is a reflection of the fact that if we have these changes, everyone is doing a little bit all the time. So some of the microbes are up a little bit, some are down a little bit, but the next day the new nutrient source comes in, a different population expands a little bit, but you're not seeing wild swings. There's sort of constant variation in the food, constant small variation in the microbial population. But it's a great illustration of a connection between diet and the microbes. So and a notion that what we are eating can be a big influence on what's happening with that gut population. Now, we can also think about this to step away from the science and get back to more of the practical everyday side of things. Eat some cauliflower, eat some beans. You know that your microbes are going to be doing things that are a bit different than normal, right? We know the foods that we eat influence our microbial population. This is trying to get at, okay, how? What are the details? What are the particular groups that are changing, and what are they doing? Again, we may feel the effects. This is trying to understand the details. So the other thing that came out of this study that was really interesting is that in some situations, some microbes responded to a particular food group. So people ate a particular group of grains, and this group of organisms increased in all people. Okay, so we now know, hey, if we wanted to influence that population of microbes, eat more grains, that population will go up. Okay, but they also found that there were a whole group of situations where there were unique interactions. One person ate one type of food, one microbial population expanded, but that didn't happen in anyone else. That happened in just that individual. So what do we do with that information? Well, it turns out that some of the variation that we see in the population actually gives us still consistent biochemistry. So what we see in the top here are the different organisms, okay? And we've got different body locations. And again, we can see, depending on where we're sampling, just based on the different colors, dramatic differences in the microbial population. Okay? Huge differences across the top. If we look at the bottom, these are different metabolic activities. So the green, for instance, is the use of carbohydrates. So if we put these together, we can see that in some situations, we've got, if we compare this part to even the neighboring area, we've got vastly different microbial populations, totally different groups of microbes. But if we look at the metabolism, they're doing the same stuff. They're using the same materials. They're producing some of the same products. So remarkably, one of the things that we've learned from these studies is that it may not be about having a particular species. It might be more, what organisms do you have that are carrying out a particular activity? So we've got a whole other layer of complexity. Great, we can identify what's where. We've got a whole list. We can compare lists. Okay, but that's not the whole story. I may have one organism that's consuming a particular food that I'm eating, a particular sugar. Maybe you have a different organism, but it's doing the same thing. We compare our microbiomes. Oh, look at how different these are. Well, except functionally, the same activities are happening. So again, how do we come up with that notion of what is the normal population? Well, it has to be more than just coming up with a list. Okay, we've got this list of bacteria. We have to look at some of the things that they're doing. And we have to recognize, again, the tremendous diversity of microorganisms. So these are 127 tongue samples. We've got samples from 127 people looking for a particular genus, Streptococcus. And if we think about the oral cavity, Streptococcus mitis is one of the organisms that contributes to tooth decay. Okay, so we care about the fact that there are Streptococcus organisms living in the mouth. And in every person in the study, they found Streptococcus. Okay, so this is definitely a group that's resident in the mouth, active, doing things. But when they looked more closely, turns out that different people have different individual species. They're all related, they're all similar, slight variations. How much does that variation matter? Maybe it matters a lot. Maybe it doesn't matter if those species are just doing the same thing, right? If they're all producing acid, breaking down sugar, and then leading to tooth decay, then maybe we don't care that there's this variation at the species level. We just care, hey, there are Streptococcus species that carry out this metabolic activity, and that's what we need to pay attention to. So we're trying to get a handle on what that normal microbiome is. And again, some things are clear cut, other things are a little murky. But something else we know is that this population can change, and it can change in response to really everyday encounters we may have. So probably one of the best known or best understood in that regard is the use of antibiotics. So we take an antibiotic to deal with a bacterial infection somewhere in the body. That drug is going to affect a lot of the organisms that make up the normal microbial population. Organisms that aren't doing anything bad to us, but are going to be affected by the drug. And we can see that very quickly in the makeup of the microbial population. So this is looking at gut populations. We've got 3 locations in the GI tract. And you can see so we have, again, no initial antibiotic treatment, and then 0, 18, 36 hours post-exposure. So just in a matter of hours, and we already start to see some large changes. Not huge changes in the upper portion, but we get down further into the GI tract, and we've had this massive change in the balance of the microbes, because that antibiotic has killed off some microbes, which then allows others to thrive. We've removed some of the competition. And so in just a matter of hours, we've got a huge change in that population, just in that one environment. We're just looking in one location. Now, sometimes that can rebalance, right? Things will find their own level again. Other times this can be a more serious problem. So something I'm going to talk about more in one of the later sections is exactly what's being looked at in this study, which is Clostridium difficile, or C. diff. One of the challenges with that is we take antibiotics for an infection, which may be a C. diff infection. We eliminate the competitors. This organism grows back faster than its competitors. And so we get a much larger Clostridium difficile population, which then maybe we treat with antibiotics again, and we cycle. Now, again, later section, we'll talk about some things that are being done to try and break that cycle, but it shows us that the microbiome is dynamic. It is changeable. It responds to changes in us. It responds to changes in outside events as well. And it varies with age. Again, it's not constant. As we change, as our physiology changes, the microbial population is going to change as well. So if our digestion is altered, then that's going to influence what materials make it down to the microbes to act on. Interestingly, something we don't think a lot about day to day is that the stomach being really, really, really acidic is important for killing off a lot of the microbes that are on all of the food that we eat. If we're not producing as much stomach acid, some of those microbes can get through and then become established in the gut. Again, situations where normally they'd be killed off, our physiology has changed, our microbiome changes. Again, we are trying to get a handle on what is a normal, what is a healthy microbiome. And we're looking at, again, not just a total average of humans, but we're trying to look at different populations, different situations to understand what's happening. So this summary is, again, from something where we're looking at those who are healthy and looking at what organisms they have and seeing how that compares to people who may have other medical conditions. Now, this is often described as what are the good bacteria and what are the bad bacteria. Life isn't that simple. E. coli is in many ways beneficial, but if it gets into the bladder, it causes a UTI. Do you describe that as a good or a bad bacteria? Well, it's good when it's where it's supposed to be doing what it's supposed to do, right? So again, we're starting with observation and correlation, and we're hoping to edge towards, okay, these are organisms we want to have, these are organisms we want to avoid, but bridging that gap is tough. But you can see, again, looking at different populations here, we've got whole lists of organisms that are changing as a function of age. So we've got organisms that decrease with age, organisms that increase with age. That's the first step. How are these populations changing? And then the next is, can we group these with other medical conditions or other changes? All right. So the last portion, I want to talk about some specific examples of how we benefit from having microorganisms. Some of these are mutualistic relationships, things where the microbe is getting something from us and we're getting something from the microbe. And some of these are simply examples of competition. Examples where having one microbe prevents another microbe from becoming established. So having microbes living with us will stop potentially harmful microbes from being able to cause infection. So our friend E. coli. E. coli is part of the gut population. Again, really well studied because it's really easy to grow and really easy to work with. In the gut, it's only a small part of the microbiome. It's maybe 1% of the colon bacteria. But that population of E. coli produces more than half of our vitamin K. So we get vitamin K from the diet. We get a lot of vitamin K from E. coli living in the gut. So this is an example where E. coli has a nice, cozy environment to live in. It has a constant supply of nutrition from all the foods that we eat, and it makes vitamin K for us. So having E. coli present is a good thing. But more than that, E. coli influences its environment. So E. coli lives in the colon, again, a really low oxygen environment. And there are lots of microbes that live in that environment because they can't handle oxygen. They'd be killed by oxygen. E. coli consumes oxygen. So it actually helps to keep that as a really low oxygen environment. So one of the benefits of having E. coli there is making vitamin K for us. Another benefit is actually shaping that environment in a way that other microbes are able to grow. So it helps to maintain that particular population. It also produces chemicals that inhibit the growth of organisms that may have the potential to cause disease. So starting to get into that competition side, having one microbe present can help to stop others from being able to grow there. Again, there's a lot going on between the microbes. If one is using a nutrient, then another isn't able to. If one is occupying that physical space, then another microbe won't be able to become established. So if we have a resident group of microbes and we consume some food that has something that might potentially do us some harm, if that new microbe can't find anywhere to settle, can't find any food to live off of, then we're going to be protected. We consume a lot of foods that we can't fully digest, fully process, which brings us back to things like beans and cauliflower. We consume a lot of plant carbohydrates that are large, complex molecules, and we don't have the enzymes to break those down, but the bacteria do. Now, a lot of that is for their own benefit, right? It's so that they can grow and multiply and do all the things that they do. But it turns out that some of the products that they produce at the end of all of this processing, we can then absorb. So we tend to focus on, we're very, very self-interested, right? So we think about digestion, it's all about what humans are doing. It's what our enzymes are doing, what our organs are producing to break down foods. But it turns out that our microbial population is doing some of the work as well. It is breaking down some of those complex materials into forms that we can then absorb and benefit from. So turns out it's not all about us. And then there's some more specific competitions. And so we've got the complicated diagram, but I think really the telling result here, the experimental result is the one that's on the left. So these are two different groups, two different species of Staphylococcus. So two different bacteria closely related to each other. In the middle, we have, the background is Staphylococcus aureus. Okay? Again, part of the normal resident population, but also with the potential to cause us disease. The circle in the middle is this second organism, Staphylococcus lugdunensis. And it turns out that that organism produces a chemical that inhibits the growth of Staph aureus. And on the left and the right, you'll notice that there's this clear area around that central growth, right? So the background growth here, Staphylococcus aureus, there's that clear zone because it's inhibited by this other bacterium. So these bacteria are competing with each other. They are doing something, one is doing something to stop the growth of the other. So you can imagine then, if we have a population of Staph lugdunensis, then it's going to be much harder for something like MRSA to become established, to stay around long enough to cause an infection. Again, lots of competition between the microbes where we're just off to the side, right? We're almost a spectator in this story. It's all about what the microbes are doing to each other. But if we have a particular combination, maybe that is actually really important for our protection. I study a fungus, so anytime I can sneak in a fungal example, I do that. So again, Staphylococcus, number of different Staphylococcus species that colonize us. Staphylococcus aureus, I've talked about before. Staphylococcus epidermidis, as the name suggests, we find living on the skin, sometimes in the nasal passages, but in particular on the skin. There are organisms, there are fungi known as zygomycetes, and they can cause a disease called mucormycosis. It's not terribly common, but when it does cause disease, it can be fatal. So this can be really serious. And it turns out that these two bacteria, which again, normally present on the skin, part of our resident population, these bacteria produce chemicals that cause serious damage to the fungal cells. And so down at the bottom, we've got these shriveled up, damaged fungal cells just through exposure to the bacteria. So the bacteria producing something that caused damage to these fungi. So if we're colonized with the bacteria, if we happen to come into contact with the fungus, it's probably not going to last long enough on the skin to be able to do us any harm, right? And this is just because we happen to have some of these bacteria present. Right, I study Candida albicans. That is what I look at. Not on the clinical side, but on the cellular side. How does this organism behave in its environment? And so in the third of these sections, I'm going to talk a lot about Candida. But there's a really great competition story when it comes to Candida albicans and bacteria. So Candida albicans is the cause of yeast infections, vaginal thrush. Can cause other infections as well, but that's the one that I'm used for this example. So in the same environment, we have lactobacilli. So we've got a collection of bacteria that can ferment and produce lactic acid. When they do that, when the bacteria are active, they're producing lactic acid, it changes the pH. Well, it turns out that a low pH affects the growth of Candida. It keeps it in the yeast form as opposed to the filamentous form. It changes the expression of some genes that are involved in adhesion to host cells. So having this mixed population maintains a balance. Okay, so the Candida is present, but it's not doing anything to provoke a strong host response. The bacteria are just hanging out, doing their thing. When the population is disrupted, things change. So one of the triggers for yeast infections is antibiotics. Taking antibiotic gives us something like we see over on the left-hand side, where the bacteria are wiped out. Now the Candida has the opportunity to grow, the opportunity to change what it's doing, and we have infection. And it's just about what two microbes are doing with each other in that particular environment. But it's also a great example where we've got two very different microbes. We've got a bacterium, we have a fungus. The bacterium is affected by the antibiotics, the fungus isn't. And so that one intervention gives us a sudden and rather large change in that microbial makeup, and then in what is happening in terms of what's going on with us. So it's a complicated system, but it's really interesting when you start to look at the huge variety of these microbes and all the changes that are happening over time. Again, some of this, this situation we've got a pretty good understanding of. The connection between some of the foods and some of the changes in the microbial population, we understand some of that fairly well. But there's a lot where we might know something about, say, what the diet is. We might observe the changes in the microbial population. We don't always know how to put those two things together. And so that's where the challenge is, but it's also where a lot of the new investigation is going. And some of the things that I'm going to talk about more in the later sections will be not just on the nutritional side, what nutrients might we get from having a resident microbiome, but what molecules are being produced that are, say, getting into circulation and affecting brain activity, affecting mood, affecting development. How do we go from just that initial colonization as an infant to growth? How might the microbial population affect how we are growing? Because something else we have to remember is not only do we have this community of microbes with us, but humans arose in a bacterial environment. We have never been without microbes. So no part of our history is free of microorganisms. And we're only just starting to understand how important that relationship is. So the last thing I'll mention is, again, just that connection between us and our environment. So this was a study that was done a few years ago, looking at the New York subway and sampling surfaces and then comparing what was found to the human microbiome. And perhaps unsurprisingly, we are leaving microbes wherever we are, wherever we contact. But it's not just about skin microbes. Maybe that's the surprising bit. When we sample our living environment, we find members of all parts of our microbiome from all different locations in the body. So we are surrounded with microbes, we are covered with microbes. And again, a lot of these organisms may be moving back and forth between us and our surroundings. Right. With that, I'm happy to take any questions. Thank you.
How does the mother impact a new infant as it relates to her biome?
Right.
You seem to maybe indicate that you get born, and in that instance you're picking up everything around you. But what is the newborn getting from the mother?
Right. So that's a great question. So what is that link between maternal and newborn microbiome? So part of it is through delivery. Part of it is just through physical contact. And so that can be direct skin contact. Part of it is also related to feeding. So some of it is going to be just direct contact. So breastfeeding will have introduction of microbes from the mother's skin directly into the mouth. But also the difference in composition between milk and formula will influence what microbes are becoming established early on. Some variation there. But another part of the story is actually about antibodies. So maternal antibodies cross the placenta and are in circulation in the fetus, and that is part of our initial protection, because we don't start producing antibodies until we are born and until that system is activated. So part of our initial protection is going to be from those maternal antibodies, but that will also then influence what microbes can become established. Breast milk also contains secreted antibodies. So that will influence, so if we have antibodies against a particular pathogen that are ending up in the newborn gut, then that will influence which organisms can establish a population there and which ones can't. So again, this is a place where there has been a lot of debate whether or not the uterus is a sterile environment. And well, right, but this is getting at this it's heading that direction. So I guess actually one thing I should have pointed out too is I emphasized all the places we have microbes, right? Truly internal environments are sterile. So the blood, there should be no microorganisms. Within an internal organ, so we looked in in the spleen, there should be no microbes. And if we find microbes in that environment, we know that there's an infection. So before birth, the fetus is in a sterile environment. There's nothing because there shouldn't be any microbes in the maternal blood supply. There shouldn't be any source there. There should be no living microbes in that environment. There's been the suggestion that there is a uterine microbial population, but I actually read a great article just a few days ago refuting that, indicating that the suggestions that there are microbes in that environment are probably contamination during sampling. So that remains. So before birth, there should be no microbes present.
All right, we have actually two comments and a question in the chat. The first comment is from Sue, who says, I ran studies in Colombia and Mexico for registration of pylori treatment for Abbott Laboratories in 1996.
Cool.
So she's joining us online today. Also, another comment, the oral cavity has a minimum of 700 species. I am surprised that makes up only 6% of the total microbial DNA. And then one question.
Yes, I have a question. I wondered if there have been any studies done on the effects of radiation and chemotherapy for cancer in the gut area, particularly colon cancer, for instance, and what impact that has. Thank you.
That is a great question, and I do not have an answer to that. I have something, I have an answer.
Would you repeat that question?
Oh, sure. So the question is what impact does say chemotherapy or radiation treatment in terms of anti-cancer treatments have on the microbiome, and in particular the gut? And I'll admit that's something I haven't looked into. I expect that work has been done. I do know, again, going back to what I study, I do know that some of those treatments which affect the immune response can allow for the development of oral thrush, again, candida infections, as well as other opportunistic pathogens. But in terms of the specific effect on the gut microbiome, I don't know, but now I'm really curious to look that up.
Just one moment,
Yep.
please.
If you take an average man or an average woman, is there a difference in gut biome between the?
Oh, is there a difference between the sexes? Oh, that is a great question, and I don't know the answer to that either.
You can just ask my wife.
So I'm going to lean back on the fungal side of things. So this isn't about part of our resident microbiome, but the fungus Paracoccidioides brasiliensis is actually estrogen responsive. So the level of estrogen in the body influences the cell shape for that particular organism, because it can grow in different forms. And so there actually is a difference in disease rates based on the estrogen levels. Again, it's not part of our resident microbiome, but you wouldn't be surprised if there were at least a few examples of microbes that were behaving in a similar fashion.
Okay, we have another question online from Laura. Are probiotic capsules recommended after strong antibiotic use, or are more natural foods, liquids that contain probiotics better?
All right. So I plan to talk more about probiotics in the third session. Because it's interesting, because again, we've talked about, hey, we see these differences, can we influence it? And there's a lot of discussion about probiotics. I will say that in terms of taking antibiotics and then taking a probiotic or eating some yogurt, that is a situation where there does seem to be a good correlation to repopulation. And so there can be some benefits there. It's an area that's not well regulated. But yes, there are certainly some benefits there. And a lot of the things that are packaged up in like a probiotic tablet are going to be the same bacteria that we find in, say, a yogurt culture. And so there may not be much difference in terms of the source in that scenario.
Okay, I've read that to allow a child to play in the dirt is a good thing. And I'll give an example just of two families I know. One is really hygienic and careful about what they do and where they go and where their child plays. And the other one is kind of like the child eats off the floor sometimes, and loves playing in the dirt. I'm just wondering if that makes sense to you as a way for a child to develop or come in contact for more microbes so that their body in the long run is healthier, can fight off infection? Because the result, what I'm seeing is the one family picking up, seems to have more sickness, and the one that's kind of allowed this bacteria play tends to be a healthier family.
So the short answer is yes. And the longer answer is it gets complicated. So so part of this is about the host and part of it is about the microbes. So, should we have lots of contact with things when we're young? Yes, because we need to train the immune system to recognize what is harmful and what isn't. It's all foreign, but what do we need to respond to and what do we not need to respond to? You can also imagine too that part of it is establishing that population, and this gets at the notion of, okay, well, what's the good mixture to have? And again, we don't necessarily know the answer to that, but it's getting at that idea. There are, when we look at some aspects of allergy and asthma, it seems like there's a connection between us not being infected by things like nematode worms as often, and sort of a misdirection of the immune response. That some of the reactions that we used to have against things that could cause us harm, if we're not exposed to those, might end up misdirected and then cause us harm that way. So that's an area that people are really looking into in some detail to try and understand. But the other thing that's interesting when you read papers in that area is everyone always goes to great pains to point out that modern sanitation and hygiene are good things. No one is arguing that this, we should stop cleaning. That, in terms of public health, broad scale infectious disease, I mean, it's night and day compared to 200 years ago. But we are starting to recognize that actually appropriate contact with some microbes is beneficial.
I read an article one time about a group of Finnish kids and a group of Russian kids that lived at fairly same proportion of nutrients and things, but the Finnish kids were well inoculated for everything, and they were much cleaner society. The Russian children were playing in the dirt and all that.
Yeah.
And the allergens, they had hardly any allergies in the Russian group, and a proliferation of many different allergies in the Finnish group.
Yeah. So it's the notion that if the immune system isn't, so allergy is most often an inappropriate reaction to something that may be foreign but isn't harmful. So we inhale pollen from the trees. The pollen can't do anything to us, but we might react as if it could. And really, that's what a lot of allergy boils down to at the simplest level. But the question is, can we be is it that we are responding to something in that fashion because the normal target for that reaction isn't something in our daily lives anymore? Again, some suggestion that maybe we can try and do some retraining or influence that a little bit. But yeah, it's the notion of the hygiene hypothesis that we need some of that exposure to have a balanced reaction.
I don't understand much about MRSA, but if you had MRSA and had an infection and they treated you, do you always have MRSA? Is it always just floating around, lurking, waiting to come back? Or
Okay, so there are a couple of things there. So one is, Staphylococcus aureus colonizes most of us, so it's easy to come into contact with it. Staph aureus also survives quite well on non-living surfaces. And so in athletic facilities, for say university students on a track team, football team, that sort of thing, working out in the gym, there can be transmission from person to person because the bacteria are quite comfortable being on the weight benches. And so given the opportunity, someone else picks it up and you can establish a new infection. So that's one thing, is it's something that we're likely to be in contact with just in general. With antibiotics, the ideal scenario is that you take the course of antibiotics, you totally eliminate the population that was causing the infection. It's all wiped out. That doesn't, again, that's the ideal scenario. You may have some resistant members of the population remain that could potentially reestablish and cause a new infection. Is that getting at what you were? Right. So yeah, so that situation is something where we've got a little more control. So do a thorough clean of the operating room or the treatment room, because we know we can wipe down all the surfaces. We know we can do a pretty good job with that. With something like MRSA, it's in circulation in the population. So when that resistant strain was first identified, it was in healthcare settings, and that was about it. Since then, we now see community-acquired infections, so we know that it's just in general circulation. It doesn't always cause infection, right? It can just be part of the normal population and spread from person to person potentially without causing any harm, any damage. But it also means like that's not something we're going to wipe out. It's always going to be around.
On one of your slides, I saw a phrase called immune signals or immunity signaling.
Right.
So I took that, perhaps incorrectly, to mean that these little critters can signal the immune system. And is that a pretty common capability across the population?
So that's, I don't know if I can answer the common side of it, but certainly there are a number of examples that have been discovered.
And is there a common, my real question, is there a common mechanism for that signaling?
No, because the goal of that signaling can be very different. For some organisms, it is trying to manipulate the immune system in a way that maybe maintains a balance, right? So that the organism isn't running riot and causing us a lot of damage, but we aren't also going after it aggressively, that it's keeping the immune response in a state where that organism can stay there and do its thing. But there are also situations where pathogens may be manipulating the immune system in a way that allow them to cause damage, to escape from the initial site of infection, that sort of thing. It can be distracting the immune system, releasing a chemical so that the immune system responds to that rather than the original pathogen. It can be a whole variety of different mechanisms.
Yeah, first of all, my hat's off to people who study an incredibly complex situation. Regarding the MRSA issue, I know, having worked in hospitals, that anybody who has ever had in the past a history of MRSA infection is now treated as having it always. And when they are admitted to the hospital, then they are immediately put into isolation, whether they have the infection or not.
Right.
And, I'm not sure always, how good that is, but there continues to be debate on that. But I suppose that in one scenario, the MRSA was never fully eliminated. It only, with the antibiotic, you only knocked it down to where the other bacteria can keep it in check. And so you continue to have it. The other scenario is that you have been exposed to these bacteria, and your exposure doesn't go away because your relatives may have it, and you're going to be in contact with your relatives wherever you got it from. You may not be eliminated. You may not eliminate that MRSA from your life. So that's part of the reason. But there's an incredible amount of waste that goes on in the hospital because of the protection, the extra gowns, the masks, all of the waste that goes on as a result of it. This
bug has really affected us in a number of different ways. Yeah.
Our time is finished. I'm blown away. I don't even know what to say, but help me thank Ian for a great session, and we'll see you next week.